All right, guys, we're just going to continue on this series with another macromolecule that you guys have to know in biology, and it's proteins. So we talked a little bit about carbohydrates. We know we need carbohydrates for energy purposes. We know that carbohydrates, when we take a look at them, there's one carbon for every two hydrogens, and there's one oxygen, so it's in that 1 to 2 to 1 ratio. But now we're going to delve into this whole idea of proteins and why we need proteins. One of the major reasons that we need to have proteins in our diet is due to the fact that they help provide our structure. So structurally, things like enzymes, we take a look at muscles, your new body parts and things like that we build all contain proteins that are in them. So we kind of use proteins as a structural property when we take a look at them. Also, when we've exhausted our energy reserves as carbohydrates and fats are concerned, our next energy reserve that we tap into are these proteins. So in this particular video series, we're going to take a little look into actual what proteins are, what they're composed of, and how we're able to build them. So when we take a look at proteins inside of our bodies and in other organisms, we see that proteins are molecules that make up everything from spider webs to parts of your muscles to the hemoglobin that transports oxygen in your blood. They have two major functions in living things. They're a source of raw material to synthesize new body parts. In order to build things in your body, you have to have some of these amino acids and proteins laying around in order to rearrange these and build parts of your body, things like enzymes, stuff like that are inside of you. And it's also going to supply energy when the carbohydrates and lipids are exhausted. So we go through our carbohydrates. The next thing we tap into is our fats, and then we're going to go into proteins. So structurally, when we take a look at proteins, they're composed of these little building blocks that we call amino acids. And we see, when we take a look at them, an amino acid consists of a carboxyl group. All right, this is our single hydrocarbon chain that we have right here. Okay, and then we have an amino group over here on this side right here. Remember, the carboxyl group is a C double bond O to OH. So C double bond O to OH. Those are my carboxyl group. This is that central carbon group that's right here that has a hydrocarbon on it. And then this is the amino group that's right here, this NH2 group that's hanging out over there. We also notice when we take a look at the amino acid, there's this new group that we have that's called an R group. And what an R group is just basically a side group, and there can be 20 different R groups that we have. So when we take a look at these R groups, there's 20 different amino acids. The only difference between the amino acids is the side group, or this R group that we take a look at. This is going to give the amino acid that we have a different charge, a different solubility. So it's going to cause it to behave a little bit differently depending upon what R group that we add out of one of those 20 that are there. We can kind of think of these 20 amino acids like letters of the alphabet. The order in which the amino acids are joined together controls the protein that they make, just as the order in which letters are joined controls the word that is made. Changing one amino acid changes the whole protein. If you start with the word warm and change one letter, an A to an O, now you have the word worm. So we change the word warm to worm. We know we're going to build something a little bit differently when we do that. So we're going to change the meaning of that particular protein as a whole when we take a look at it. So that's kind of a key feature of these actual proteins is this R group. I'm going to show you the different 20 R groups that we have. And we can kind of look at what they look like when we take a look at these 20 different amino or side groups or R groups. This is just a picture, guys, that you're taking a look at, and you can see the different R groups that exist out there. These are the 20 different R groups, and these are the 20 different amino acids that we have. So we can have a variety of combinations that we can have joining these particular amino acids together to build a particular protein. What makes each amino acid different from one another, like I said, is these side groups or these R groups that I have circled in green in this particular diagram itself. If one has a positive charge, the other one has a negative charge, they're going to kind of attract each other, and that's going to cause a bending inward of the molecule. If they're both the same charge, it's going to cause a bending outward of the molecule. If they don't have a charge at all, they're going to kind of stay neutral when we take a look at them. So this allows the protein to have a particular shape and a particular structure when we take a look at it, depending upon how this R group behaves in the protein molecule itself. So on that document, it's asking you to draw two amino acids side by side. So this is what that would look like if you're going to draw the two amino acids side by side. When we take a look at them, like I said, this is the amino group, this is that side R group, and then this is that carboxyl group that's up here with that C double bond O to the O, to the C double bond O to the OH is what you have there. So when you draw that, that's what you're going to have on your paper for the protein activity. It would look something like that. So if you go back to your paper, we can see that the bonds that hold amino acids together are called peptide bonds, and they're formed by a process called dehydration synthesis. We studied dehydration synthesis, guys, when we took a look at the carbohydrates, and we know that when something's dehydrated, that means that it's lost water. So dehydration means that it's lost water. So it means that it's lost water. What are you doing if you synthesize something? When you synthesize something, synthesize means that you're going to make something, to make. So if we're going to hypothesize, what does it mean to go through the process of dehydration synthesis? That's going to mean that we're going to lose water, so loss of water, to make something. So in this case, we're going to lose water to be able to join these two amino acids together to help build an actual protein itself. So when a peptide bond forms through dehydration synthesis, the hydrogen from one atom group of one amino acid combines with the OH of the carboxyl and another amino acid. This forms a molecule of water. So it asks you, when you come down here, it says put a circle around these particular atoms in the diagram itself. So we take a circle, we're going to put around these atoms in a diagram itself. So what it's asking you to do in this previous question is it's asking you to put a circle around the hydrogen atom, and it's asking you to put a circle around the OH atom that's here in the carboxyl group. And when these two atoms combine, it forms water. So we've gone through this dehydration synthesis is what's happened. So I've gone through here. There's H2O that's been removed. And you can see right here now, this carbon now is going to bond with this nitrogen that's right here. And this bond that's right here, is what we call the dipeptide bond. So that's where your dipeptide bond is going to be. On your test, I'll ask you to show me the dehydration of an amino acid, and you have to find the OH group and then the H group. In some of your biology textbooks, guys, you'll kind of reverse this image. So rather than say NC, you'll say CN here, but it's still a dipeptide bond that you guys have to know. So moving back to our paper that we have here, we just completed the dehydration synthesis to bring two amino acids together to form what's called a dipeptide. So three or more amino acids joined together is going to be called a polypeptide. So this is going to be polypeptide. In this particular question here, it's asking you to identify the peptide bonds that are here. Remember, the peptide bonds are when we, from the previous part right here that we took a look at right here, you can see this is going to be the peptide bond. So where this C bonds with the N here, or this N binds with the C here, is going to be that peptide bond. Note it's going to sit in between the two R groups when you look at the peptide bond. So this peptide bond, there's a peptide bond right here, there's a peptide bond right here, and there's a peptide bond right here that we have. So that's the peptide bonds that we have in this particular molecule itself. And again, this is a polypeptide that we have. We take a look at proteins. Proteins are polypeptides about 200 amino acids long. So we take 200 amino acids, we join them together, and then we have an actual protein itself. When we take a look at these amino acids, remember there's 20 different amino acids depending upon the R group that we have. The R group's going to behave differently depending upon the other R group that's beside it that we have. So for instance, if this R group here is positively charged and this R group here is negatively charged, basically opposites are going to attract. So what this amino acid is going to do is it's going to fold in on itself is what it's going to do. So if this R group is positive, this one's negative, then this whole entire molecule is going to fold in and kind of give it that particular shape itself. Now, if this one was positive and this one was positive, they'd repel each other. So they'd repel each other and they go further away from each other. So depending upon the charge of the actual R group itself, it's going to give the behavior or the shape of the molecule its critical characteristic that it has. So if two positive side groups are close together, what are they going to do? They're going to repel each other. And it's going to have it's going to repel each other. So this molecule is going to have a characteristic shape. You'd have a positive charge here, a positive charge here. They're going to go further away from each other. If a positive side group and a negative side group are close together, what's going to happen is they're going to attract each other. So they're going to attract each other is what's going to happen in that particular case itself. And what that's going to look like is, so I have a positive charge here and a negative charge over here. Opposites are going to attract. So this molecule is basically going to fold in on itself is what it's going to do. So it's going to fold in or come closer together is what's going to happen in that particular case. Is it's going to fold in on itself is what's going to happen when we take a look at these opposites attracting. If the side groups have no charge, then they're not going to repel, they're not going to attract each other. The molecule is going to stay in the same shape, so it will stay the same. It won't repel, it won't attract, it will just stay neutral. So we'll just say, if we have a no charge going across them, it'll just kind of stay the same. It'll have that same flat line appearance that it has there. After you guys got done with that, then you guys built the actual molecules themselves. You joined the amino acids together, is what you guys were able to build in class. And you guys did that in class with the modeling kits. So the final thing that we have to do is we have to just kind of go through the practice to make sure that we're right. This one's asking us to draw the structure of an amino acid. So remember, we have this carbon, this central hydrocarbon that's bonded to an R group that's going to be down here. And then off to the side here we have an amino group. And then we have our carboxyl group that's here. How many different amino acids are there? Well, there's 20 different amino acids, because there's 20 different R groups that we have when we take a look at them. Amino acids are joined by a process of dehydration synthesis. And the bonds between them are called peptide bonds. True or false, changing the order of an amino acid in a protein makes no difference. That's completely false. Okay, if I change the recipe that I have for a cake or something like that, this cake's going to taste differently. Same thing, if I change the order of these amino acids, there's 20 different ones. If I change the order of them, you're going to build a completely different protein that you're going to have there. So that's kind of what you're going to put here, is that it does have an effect. You can kind of give the baking of a cake. It's changing the complete recipe of what you're building. So you're going to build something completely different. Two amino acids joined together are a dipeptide. 3 or more amino acids joined together are called a polypeptide. So that concludes the macromolecule of proteins. If you guys have any questions, don't hesitate to see me in class or ask. It's kind of easy molecules once you kind of understand the process of dehydration synthesis. Knowing that there's 20 different amino acids, the order of these amino acids is going to change the shape of the molecule and what you're actually building itself. Like I said, on your test, guys, you'll have to know the carbohydrates, the proteins. Next, we're going to talk a little bit about lipids. So if you look at the next video series, we'll take a look at the next major molecule, which is lipids as well.